It's really a great pleasure for me to be here again, and it's also for my wife. I've been a frequent visitor to Taiwan in the past. It's not my first visit, but every one, I can assure you, was the most pleasant one and interesting one. So what I'm going to talk today is a story which interests us since quite a number of years, and it's mainly based on epidemiological observations. When we look today into the situation of infectious agents in human cancer, we can roughly calculate that about 21% of the global cancer incidence is linked to infections. These are not only viral infections, but also bacteria and parasitic infections. We can roughly calculate that about 2/3 of those infection-linked cancers are due to viral infections, and about 1/3 to Helicobacter pylori in gastric cancer, and about 1% to parasitic infections. And here, in particular, in southeastern Asia, in southeastern Thailand and China, Opisthorchis infection, liver flukes infections play a significant role in liver cancer, as do schistosoma infections in Egypt in bladder cancer. Now, the discovery or the identification of infectious agents playing a role in human cancer paved the way for preventive measurements of cancer by preventing infections. And this is true by pioneering work which has been done here in Taiwan concerning the hepatitis B virus-linked cancer prevention, which clearly has been demonstrated by now to be a cancer preventive vaccine if children are vaccinated during the first year of life against hepatitis B, usually being infected by mothers who carry this virus persistently for a lifetime. It became true for those agents which we found during my time in Freiburg, namely HPV 16 and 18, where vaccines exist right now which prevent the precursor lesions of cervical cancer. The time periods which elapsed until cancer develops from primary infection is usually something like 15 to 30 years. And the vaccines in many countries have been introduced or licensed in the year 2006. So it's at the moment still too early to state that cancer is also prevented, but it's almost certain that it will be prevented as well. In addition, let me just point out that, of course, the treatment of Helicobacter pylori infections by antibiotics, of parasitic infections by chemotherapy, I don't see it here, by chemotherapy also eliminates those types of infectious agents, but it does not leave an immunity afterwards. And so indeed, the cured patients from this infection may become reinfected afterwards. Now, in the case of vaccination, we know that we have a long-lasting effect, and it became also possible to try to develop a prevention by therapy of persistent infections. That is particularly the case for hepatitis C virus infections and also for HIV infections, the AIDS virus infections, where treatment presently is reducing the load of the respective agents and seems to prevent as well Kaposi sarcomas and B-cell lymphomas linked to long-lasting immunosuppression. And in the case of hepatitis C virus, apparently, at least for the time period which is available right now, of something like 4 or 5 years, it seems that the elimination of these types of agents by therapy probably will represent in the future a kind of secondary prevention of cancer as well. Now, the question which interests us since a very long period of time is the one whether besides those 21%, are there existing additional cancers which might be linked to infectious events. And this list is a relatively old one, which I compiled about 7 years ago, about those where we can raise suspicions that infections may be linked to cancers, mainly to cancers, first of all, occurring at increased frequency under immunosuppression. I don't know, can you hear me well? Or is it better? No, this doesn't seem to work. Okay. Cancers arising under increased frequency under immunosuppression. Of course, we do know that most cancers which occur under immunosuppression have some links to infections, but there are clearly others, like kidney cancers, thyroid cancers, and a couple of others, which are also increased under those conditions, and yet have not been linked up to now to immunosuppression. I will not dwell on this question in the subsequent talk. So the next point is of quite considerable interest for us, namely cancers with reduced incidence under immunosuppression. And here the outstanding example is really human breast cancer. Prior to the therapy of AIDS infections, there was an approximately 15% protection in breast cancer incidence of those patients who suffered from HIV infections and a severe immunosuppression. Right now, due to the HAART therapy, the highly active antiretroviral therapy, this effect seems to be abolished. But clearly there's no increase under those conditions. I will come to this point later on again. Then there's the cancers influenced by other basically non-tumorigenic infections, which is certainly true for cancers which occur during the first 2 years of life, particularly B-cell leukemias, neuroblastomas, and brain tumors, where infections, non-specific type of infections, basically all or the majority respiratory tract infections, have a protective effect against the incidence of those types of cancers. And this has some interesting implications. Again, today I will not talk about them, but this clearly may point to something which may occur prenatally in those in the mothers who deliver the baby subsequently. Now, the main point of my talk today will be nutritional cancer risk factors possibly linked to infections. And the idea originated from observations which have been made over a long period of time, namely that we do know a number of human pathogenic viruses, in particular the polyoma viruses BK and JC, widely spread in human populations. About 60% of all adults carry them consistently and latently, which do not cause, as far as we know, tumors in humans. But upon transmission into animal systems, here, for instance, hamsters and owl monkeys, they are suddenly tumorigenic in species in which they cannot replicate. As they are replication incompetent, they become tumorigenic. This also counts for adenovirus type 12, 18, and 31, to a much more limited degree to Epstein-Barr virus and high-risk papillomaviruses, because they are, the latter two, are in some instances tumorigenic in humans as well. But I will mainly attribute this discussion to BK, JC, and adenoviruses. And this raised the question, is there something similar in animals, particularly those animals with which we live in close neighborhood, listed here on the upper left panel of this picture, that they may contain viruses latently, persistently, sometimes excreted in their respective products, which in the native species do not cause tumors, but upon transmission to another species, in this case to humans, in which they cannot replicate, where they are replication incompetent, is it possible that they become suddenly tumorigenic? So from that point on, we started to look carefully into the question, which kinds of cancers may be of interest. And here clearly colon cancer was standing out to some degree, because there are a large number of reports of a higher incidence of colon cancer in persons who consume for long periods of time the so-called red or processed meat, air-dried, smoked meat, something like this. Usually the increase is about 20 to 30%. Slightly more than 100 follow-up studies have been published claiming this. I found 2 or 3 which did not come up with increased data, but by far the majority had the same data. Very remarkably consistent for epidemiological studies. Secondly, countries with the highest rate of red meat consumption, and some are listed here, Taiwan belongs to them as well, commonly reveal a high rate of colon cancer. Now, since 1978, when Sugimura and his colleagues in Japan reported that cooking, frying, barbecuing, roasting meat leads to the formation of chemical carcinogens, specific chemical carcinogens, in particular aromatic hydrocarbons and some of the others listed here on the slide, the question seemed to be partially solved, because that would be an easy explanation why there is an increased risk after consumption of this type of red meat, if there wouldn't be a few things which did not fit too well. Well, first of all, it seems that meat consumption in general emerges as a specific risk factor for memory cancer. Colon cancer has not been very well investigated in carnivores. These types of cancers are extremely rare in cows, mares, ewes, goats, and sows. But on the other hand, they are frequent in female dogs, female cats, but they have also been found in other carnivores, in wild living carnivores listed here, which seems to point to a specific risk in those animals who are carnivores and consuming meat. Now, there are some other aspects which came up which are quite interesting, because if you roast and barbecue, broil, or fry white meat, poultry in particular, or fish, the same chemical carcinogens are being produced as with the red meat, sometimes even in higher concentrations as in red meat. And yet, all the epidemiological studies for fish even demonstrate a slightly protective effect for long-term fish consumption. For poultry, at least there's no elevated risk for this type of cancer noted. Now, if you look into the geographic pattern of colon cancer, there are some regions of particular interest. Of course, Europe here, Russia, and also the eastern part of Russia, they're all relatively high-risk countries. The United States, Argentina, in South Africa, particularly the white population, in Australia, in Japan, and in Korea. These are all high-risk areas, including Taiwan as well. Now, if you look into other parts of the world, it's quite interesting to see some diverging patterns. The most interesting one is Mongolia. Mongolia, which has in general a high rate of cancers, has one of the lowest rate of colon cancer. It's supposed to be one country with the highest rate of red meat consumption, and yet the rate is very low. India, particularly interesting because the Indian population, the Hindu population, do not consume beef at all. They consume mainly poultry. If they eat meat at all, many of them are vegetarians. Very low rate of colon cancer, comparable to Mongolia. Bolivia, a country which has a high rate of beef consumption as well, but a different type of beef than the common dairy beef in most of the high-risk countries, namely Zebu type of beef, has a low rate of this cancer. And Equatorial Africa has also a low rate of cancer. The cattle which are being kept there are virtually all of Zebu origin. The Watusi cattle are clearly contain Zebu genes. Now, if you look into the red meat consumption, there's some estimates about the consumption of meat. You see that the picture differs from what I showed you before. Parts of particularly North and South America are quite high consumption of meat, but also some countries I mentioned Mongolia a moment ago, but also in the west of Mongolia, France has a very high consumption, and Botswana in Africa also has a high consumption. So that's quite different from the picture of colon cancer on the other hand. Now, particularly the Indian data suggested that it might have something to do with beef consumption. And so we tried to find out which type of beef is being consumed or which type of meat is being consumed in various countries. And to make a summary statement here, wherever the risk is high, there's a higher prevalence of consumption of dairy cattle, the Eurasian type of cattle. And wherever there's a low rate of cancer, seems to be maybe the zebu derivatives, the Brahmanian type of cattle, Batusi cattle in Africa, which are all have the same origin. India, of course, has beef not consumed at all. And it's interesting to look into the genetic composition. Here the black dots, the black circles indicate the prevalence of mitochondrial genes of zebus, and you can see it here in Bolivia, really a very high prevalence of zebu genes, virtually exclusively zebu genes, part of Brazil, and also in Southeast Asia. Africa has a very high involvement of zebu genes, whereas, for instance, in Europe, the admixture of zebu genes is extremely low under those conditions. Now, it's interesting to look into the development of colon cancer in various parts of the world, and this shows some pictures, for instance, here on the left panel in Scandinavian countries, where over the years since 1975 up to the year 2010, there was a gradual increase in colon cancer, whereas in Asian countries, the situation is quite different, and America. In the United States, there's a decline of colon cancer recently, and according to my US colleagues, I was told that this is mainly due to colonoscopy, which is carefully conducted in the United States. Removing the precursor lesions means that cancer is being prevented, so the decline seems to be due to a higher degree of secondary prevention. But in other countries, particularly in Japan and Korea, the situation is strikingly different, because in Japan you can interpolate it probably starting by the year 1965. There was a rather dramatic increase in the rate of colon cancer. In Korea, the same happened. If you interpolate it, it's probably also about 20 years after the Korean War. And you can see here in the lower panel what happened in these countries in terms of meat consumption per capita. And it shows that, for instance, since the year 1970, there was a rather dramatic increase in meat consumption in Japan, and about 10, 15 years later, the same in Korea happening, now even exceeding the rate in Japan at the same time. So in a way, that seems to correlate relatively nicely with the meat consumption, whereas India remained low during this period of time. This is really the graphic demonstration of the same picture which I showed before, showing the increase in Japan and Korea. And what happened in these countries? Well, in a way, it's interesting to note, and probably many of you have experienced it, either here or in those countries, that indeed some specific meals became relatively popular. The sashimi, previously provided as fish, rare fish preparations, now is often offered as rare beef meat preparations. The shabu shabu, meat fondue, usually you dip it for a minute or so in boiling water, the slices of beef, take it out. The outside is usually well done, the inner part is still rare. It became very popular in Japan, and as I understand it, also in other parts of Southeast Asia. I do not know how it is here in Taiwan, but I presume that you have it here as well. In Korea, there's a Korean yukho, I hope I pronounced it correctly, a kur tartar, which is being served with egg yolk and some vegetables. Again, a meal which became popular in Southeast Asia and many other parts of Southeast Asia as well. In South Africa, particularly the white population, were consuming frequently biltong, which is air-dried beef mainly. Also biltong of other species, but clearly beef is the most prevalent one. So being a virologist, of course, rare materials are good preservatives, and air-dried even better preservatives of infectious agents if they exist in those instances. This shows the colorectal cancer incidence in Southeast Asia. You see on the first three slides, on the first three parts, the incidence in Mongolia, in China, and in Taiwan. You see Taiwan belongs to the high-risk countries, as well as Japan and Korea, Singapore, Brunei, whereas in many others of the Pacific islands the rate is relatively low. And if you look into the data which are published on beef consumption in China, it's interesting to note, going back to China here, it has a relatively low rate of colon cancer, that the vast majority of meat which is being consumed is obviously pork meat, supposed to be 80% of the meat which is being consumed. Beef is also consumed to about 20%, but beef of different species, namely of the Chinese yellow cattle, which are again zebu derivatives, the yak meat, particularly in regions close to Tibet, water buffaloes in the south, and also in recent years more dairy beef as well. It will be interesting to see how the developments will look in 10 or 20 years from now. What about Mongolia? In Mongolia, beef is apparently consumed to about, or consists 40 to 50% of the meat consumption consists of beef, but these are different types of beef. Namely, first of all, yak plays a substantial role, but also crossbreedings of a different subspecies of taurines in Mongolia, which have been interbred, particularly with the Chinese yellow cattle, the zebu derivatives, which is being consumed by about 40 to 50%, mutton and goat, 30%, camel meat, and horse meat. These are estimates, of course, but they seem to basically to provide some realistic figures. Meat is most frequently grilled, barbecued, and or consumed after air drying. The Mongolian boards seem to be quite popular there. I've never been there myself, so I've never experienced this. In South Africa, the braai here on the right-hand side is a very popular meal where the meat is being prepared so the inner part is still relatively rare. And if you take a roast beef, as it is shown on the left part, the inner part is still relatively rare, and measure the temperatures is up to 40 to 50 degrees centigrade temperatures in which many viruses happily survive without any loss of infectivity under those circumstances. Now, it has been suspected recently that some bacteria play a role in the microbiome of colon cancer patients, and these are in particular Fusobacterium nucleatum and Streptococcus bovis. And the reasons for suspecting it, that they have been relatively frequently being found in colon cancer in general. Both of them are also found in any other kind of inflammatory reactions, dental plaques and others. And they obviously are involved in causing inflammation, oxygen radicals, and they're probably mutagenic at the same time. As I will tell you in a moment, they may interact really with some other agents by leading to mutations in host cell DNA. So this led us to develop a kind of a tentative scheme how colon cancer might be caused under those circumstances, assuming that you have here an average lifespan of about 80 years, that the consumption of raw and undercooked red meat would lead to some kind of an infectious event during lifetime and lead to transient or latent infection by a putative carcinogenic agent. I wrote virus, but I would be more careful saying agent here. And that, on the other hand, due to the fact that we do know that not a single infection exists which is linked to cancer, which does it by itself, they always require some additional modifications being be it genetic or epigenetic modifications within the same cell, that the consumption of cooked or cured red meat due to the chemical carcinogens are being produced leads to mutations in host cell DNA by these chemical, or in the case of Fusobacterium and Streptococcus, biological mutagens, acquired genetic modifications which synergistically interact with some kind of latent infections, and that inherited genetic modifications would basically exert the same type of effect under those conditions. Now, before going on, let me just make a brief detour to breast cancer. There are a couple of studies which also imply an increased risk after red meat consumption for breast cancer. Some of them are listed here. The data are not as consistent as for red meat. There are also a number of conflicting studies at the same time, which have been published. And interestingly, there are also a couple of data on lung cancer. Again, they're not consistent in the same way as for colon cancer, and a few other cancers which I'm not going to discuss here right now. Now, if you look into the geographic epidemiology of breast cancer and colon cancer, there are some remarkable consistencies, some remarkable correlations. Let's take, for instance, the northern part of the United States and Canada, Asia, Europe, for instance, and Australia, and a few more countries. But there are also some remarkable consistencies in low-risk areas, and here specifically of Mongolia and also of Bolivia in those instances, which require some explanation. There are a few other aspects which need to be discussed in a moment. Why is breast cancer so interesting? Breast cancer is interesting because we know an animal model, the so-called murine mammary tumor virus model, discovered by John Bittner in 1938, who showed that there's an agent transmitted via the milk of feeding mothers to the newborn babies, and the agent settles down in the pious patches of the intestines, infects T and B lymphocytes there, and leads to the formation of a super antigen. The cells virtually swell. They are eluted into the blood and lymph stream, and those cells, the super antigen producing cells, produce simultaneously large quantities of viral particles. And the dose of the respective particles seems to be a responsible fact that it reaches in sufficient quantities, the target tissue, the mammary gland, becomes inserted into, usually in fragments, into those cells. Some of the integration sites have been carefully mapped and are listed here on this slide. And apparently, due to some of the hormone-responsive receptors which these agents have, particularly during lactation, they are activated and lead to the formation of mammary tumors under those circumstances. So, is there a similar agent found in humans? Well, there are at least a few claims that they exist, but other laboratories, and that includes ours, could not confirm those types of reports. Under immunosuppression, the super antigen producing cells are being suppressed, the virus dose is reduced, and under those circumstances, the risk for developing mammary cancer is also reduced in mice. That's an interesting parallel to what I told you initially about the reduction of human breast cancer in non-treated AIDS patients in the past. Now, looking into the comparative aspects of colon cancer and breast cancer, there are some inconsistencies in the pattern, particularly as far as Japan and Korea are concerned, because in the period where in Japan and Korea the colon cancer increased dramatically, the increase in breast cancer is there, but it is much more moderate under those circumstances. And in India, the increase is also there, but it is clearly higher here than in the case of colon cancer. So this is difficult to compare to the situation or to the assumption that the same agent might be responsible. the same agent might be responsible for breast and colon cancer, but it led us to the speculation that there might be the same carrier for an agent which might be linked to both types of cancer, but that this agent in breast and colon cancer should be rather different and not identical. Now that, of course, led to the consideration whether milk could play any kind of role specifically in breast cancer. Now these are the data on liquid milk consumption per capita in 2011, and you see that China, for instance, has a very low rate of milk consumption at that period of time. South Africa, Japan and Korea have also relatively low rates, but other countries, the United States, Australia, United Kingdom, they have a high rate of consumption. The milk production in India, these are data from the National Dairy Development Board in India, shows that there is indeed, since 1980, a rather substantial increase in milk production. Indeed, dairy farms have been introduced during this period of time. The breast cancer incidence is still relatively low in comparison to the consumption of milk, but it is obviously increasing, and it will be interesting, since we usually speculate that something like 20, 30 or 40 years elapse between a primary event and the development of these tumors, how the situation is going to develop in the future. In Mongolia, there's also some increase in milk consumption, not so dramatic as in other countries. It's starting basically since about 1980, 1990, and led to about, about maximally 50% increase in milk consumption in those conditions. Now, if you look into the breast cancer incidence in Mongolia, it's quite interesting to see that in the urban areas, shown here in the upper panel, there's indeed a slight increase in breast cancer. In the rural areas, there's no significant increase measurable during the same period at all, up to the year 2005. Interestingly, dairy products have been introduced to a substantial degree, particularly in the urban areas, and that might be related to the increase observed here. This shows the dairy product consumption in China, and here you can see that the milk production since about the year 2000 increased dramatically in urban areas, but it is still very low in rural areas. And as I told you before, the breast cancer incidence is still relatively low in China. Now, there was an interesting publication recently from Sweden where the lactose intolerant people were compared to relatives, which are all found in the Swedish Cancer Registry, by comparing about, 23,000 lactose intolerant persons to their siblings and parents, which are lactose tolerant, rather perfect controls in some way. And under those conditions, it was noted that in lactose intolerant persons, there was about a 21% decreased risk for breast cancer, and more difficult to explain, and about 45% decreased risk for lung cancer. I tried to find whether there's less smoking in lactose intolerant persons in comparison to others, but I couldn't find any significant data along those lines. But these are clearly interesting data, since indeed lactose intolerant persons usually reveal a reduced or no uptake at all of dairy products under those circumstances. Let's come back for a moment to the breast cancer incidence, here particular to China and India. Thailand is also relatively low, and a few African countries. And Taiwan is almost in the order of Singapore, so it's relatively high. It's not listed here in this one. But you see there's a remarkable difference between these countries and some of the high-risk countries globally. Bolivia is supposed to be the country which has the lowest consumption of milk. They keep cattle which are almost exclusively of Zebu origin, which might be related again. So this was our speculative model now for for breast cancer, which is quite similar to what I showed you before for colon cancer, with one remarkable difference. Namely, we would suspect that every person, or virtually every person, is exposed early in his or her life. But on the other hand, that acquired genetic modifications, similar to what I pointed out for colon cancer, or inherited genetic modifications, would play a similar synergistic role in eventually activating latent agents and leading to, initially to the precursor lesions and subsequently to cancer. If this model is correct, it has one important consequence. It means that cohort studies to identify dietary risk factors later in life, more than 20 years of age, and virtually all statistics which are available right now have been conducted in adults, that they will be unable to identify dairy products as risk factors if you don't include pregnant women or children during the first years of life. And so you will have still to wait for 40 or 50 years before you finally will get significant results. But in a way, it could explain at least the discordant data which have been published on breast cancer in comparison to colon cancer, but this would also lead to synergistic interactions. Well, this led us, and Esme Michel is going later to talk about it in detail, to the attempts to isolate potentially agents, putative agents, from these types of animals, specifically from dairy cattle, which may play a role in these types of diseases. And indeed, as you will hear later in more detail, a number of such agents came up. They were isolated from blood of 120 dairy cattle, which we obtained from the university, the veterinary station in Leipzig, in Germany, and also from milk, from commercially available milk preparations right now. And all of these agents, which are shown here, belonging to several groups of single-stranded circular DNA viruses of small sizes. What we do know right now is that we did not find them, at least those groups which we found in cattle milk and in blood. We did not find those in the blood of healthy human blood donors. But we did find them in 2 samples of multiple sclerosis directly, although a couple of other samples were negative for them. I will not dwell on this in any further detail, except mentioning the fact that indeed those agents upon transfection into human cells are able to be genetically active, to induce specific messenger RNAs, which are usually polyadenylated, and which is interesting because some of them share at least a portion of their genomes with bacterial plasmids and phages, which of course initially raised the suspicion that they may be bacterial contaminants, but due to the fact that they are actively expressed in human cells and the polyadenylated RNA seems to clearly point out that they are adapted at least to mammalian cells in the meantime. Well, I think I will skip this one because it will be mentioned later on the active transcription. But let me just finish by saying a few words about multiple sclerosis. 2 lesions contained 2 forms of single-stranded DNA, and one of them is closely related to an isolate which was originally described by a group in Yale, Manuelidis, and she called it Sphinx agent, an abbreviation for something which I'm not presenting to you right now. And one other biopsy and one MS serum contained a Psychobacter-related plasmid DNA as well. Now, in view of the heterogeneity of the healthy cow blood isolates, we anticipate that several different agents may be involved in different MS lesions. Some analogy exists to human papillomaviruses and cervical cancer, which clearly suggested at that time the existence of high and low risk infections, and this could be related to multiple sclerosis as well. We do know that there's a couple of risk factors which have been identified in multiple sclerosis. One, interestingly, is cow milk consumption in childhood. a couple of reports, particularly the last part of the past century, but also relatively recently, that cow milk consumption is a risk factor for multiple sclerosis. I didn't find a single report which was conflicting with this observation. Vitamin D deficiency later in life, and reactivation of latent herpes virus infections. Here specifically Epstein-Barr virus, human herpes virus type 6, and varicella zoster virus. These appear to be the main risk factors for the development of multiple sclerosis. And that led us to develop some kind of a model how multiple sclerosis could be initiated by these types of events, by postulating that in the early period of life, the early postnatal period, infection occurs by a bovine milk factor with single-stranded DNA agents, which all persist, at least as far as we know, episomally. There's no evidence for integration into host cell DNA. That at the same period of time or later on, latent infections by different herpes group viruses may occur, and that the latent infection leads to infection of individual brain cells, but rarely also to a double infection of the same cell by both the bovine milk factor and the herpes virus. And in cells which are simultaneously infected by those types of agents, the herpes viruses all tend to become reactivated from time to time under certain circumstances. And we noted in our laboratory a long time ago that indeed TGF-beta, tumor growth factor beta, is an active inducer of Epstein-Barr virus, for instance. And there's an interesting reports which claim that episodes of low sun exposure lead to vitamin D deficiency, and under vitamin D deficiency, an upregulation of TGF-beta occurs. So we suspect that this is a triggering event for increasing at least for Epstein-Barr virus reactivation. We do not know it for other types of herpes viruses. And interestingly, also something which we had observed quite some time ago is that herpes viruses in general, any type of herpes viruses, as well as adenoviruses, are able, upon infection of a cell which contains either single-stranded or double-stranded small DNA viruses, leads to a rather remarkable amplification of the latter. And this amplification leads at the same time to an inhibition of the herpes virus replication. So we assume that in cells simultaneously infected by, here for instance, EBV and the bovine milk factor, that a high amplification of the bovine milk factor occurs, infection of surrounding cells happened, and an initiation of EBV replication is inhibited under those conditions. Not the initiation, but the replication is inhibited subsequently. And this has a consequence due to the immune reactivity to the antigens exerted by the bovine milk factor to inflammation, destruction of the affected cells, and the onset of this type of disease. It's a scheme and it's a model which needs to be proven, of course. But if true, it will have a number of implications. So in case that this type of hypothesis is correct, then we can state that almost every one of us became infected with this putative bovine milk factor during the early period of life. But that's a persistent infection by BMF of the respective target cells, in this case of brain cells, remains asymptomatic. And that a dual infection of the same target cell by BMF and the herpes type virus should be a rare event, is a rare event, and only in cases of reactivation of the latter, of the herpes virus, gene expression and amplification of BMF would occur, resulting in focal spread, inflammation, and destruction of the affected cells, and the reactivation would be facilitated by vitamin D deficiency and enhanced TGF-beta activity. So in a way, some reports tried already to link vitamin D deficiency and particularly Epstein-Barr virus reactivation also to breast and colon cancer, although the data are scarce and clearly not fully convincing yet. But the identification of the responsible BMFs would not increase the present existing risks for breast and colon cancer and multiple sclerosis or other types of cancers, but would provide new opportunities for prevention, risk assessment, and hopefully in the future also for therapy of these types of diseases. So it remains a very interesting and important issue, and this should be really an encouragement, and is presented as an encouragement, particularly for young researchers, to look more carefully into the question. It's not as simple as we assumed in the past, that a single agent after infection and long latency periods is causing cancer. It always needs some other interactions. So these are the persons who very actively collaborated in this study. As Michel Devilliers was going to talk later on these agents more specifically, and a couple of devoted postdocs and technicians who are working with us on the same topic. And I'm usually showing subsequently our involuntary co-workers. Here, these are the cows from which the blood has been taken, which served so well as the source for the infectious agents which we presently have here. So thank you very much for your attention, and thank you once again for the invitation to come here.
The lecture is open to discussion now.
Thank you, Dr. Hauser. It's a wonderful talk. And I have the question because you show there's the subspecies-specific origin of the beef. So, but the beef already going to the genetic modification that we already breeding for different area of the world. And they still show the correlation between the beef genetics and the cancer incidence. So does that mean that if there's the infectious agents, they are related to the genetics method of the beef?
Well, what we can show at the moment are only the epidemiological data, which seem to point to a low risk of cattle breeds of zebu origin and also of yak origin. In this specific case, and probably also water buffalo origin. But unfortunately, we didn't have a chance to get any materials from those cattle so far. So we don't know whether there are differences. I mean, we got the promise to receive sera now from zebus and also from yaks, but we don't have them yet in our laboratory. That will be interesting to analyze it more carefully. As far as specific cattle breeds of dairy cattle are concerned, I don't know. We don't have any evidence because we looked only into one species so far, one breed so far.
Thank you.
You identify circular DNA, single-stranded. You identified double-stranded, so indicating it's a replication.
Not that we have directly demonstrated it. Indeed, we can demonstrate right now the expression of messenger RNA in human cells, and this has been mapped by now. And we also can demonstrate protein production in human cells, but we have not yet clear-cut evidence of replication. Maybe we don't even have the right cell types to replicate the agent in so far.
So how do you propose its persistence if it's not episomal or if it's not integrated into the host?
We propose an episomal persistence in this case. I mean, so far no evidence exists for integration at all. You may ask the question really, why hasn't it been discovered in those types of tissues right now by deep sequencing? The answer seems to be straightforward right now, why it hasn't been discovered. First of all, because those attempts have been made with chromosomal DNA in order to get rid of the mitochondrial DNA as well, which disturbing the reactor. Secondly, because exotic sequences, particularly if they show some similarity to phages, are dismissed as bacterial contaminations. And the third reason, which might be the most important one, is that apparently the tagging of small pieces of DNA works well with double-stranded DNA, but doesn't seem to work well with single-stranded DNA. So that is another point why it has been not found, except for very few recent publications which observe TT virus DNA also in colon cancers.
唱歌也能发生奇妙的事。在大竹国小，内向的小朋友只要一唱起歌仔戏，立刻就变成活泼有自信的人。他们将运用巧思，结合传统与现代，带给你一场最有意思的歌仔戏演出。每周六下午4点，戴文金娜赞好戏上场喽，千万不要错过哦！
飞鱼卵在这个季节哦，5、6月、6、7月这个季节的话，它就会产在海藻上面。
那西瓜在夏天我真的很难想象说，夏天如果没有西瓜，我们应该怎么办才好？
回来了，早鸟6折，套票3月9号开卖，只有3天。快上儿童影展网站查询。
各位朋友，大家好，我是侯丽芳。说起来，我跟公视的渊源非常的深，感情也很浓。公视播出了《时代之歌》怀念金曲演唱会，也是我主持，然后获得了很多的好评。诚挚的邀请各位跟我一起来支持公共电视，我们的捐款专线是02-2633-9922。
你们这个是不实指控啦！公司哪里有逼退任何员工啊？
公司已经有50几个人离职了呢。
他们都是自愿的，都有签这个自愿离职同意书。
拢是你啦，讲什么都爱跟公司什么共体时间，咱挺公司啦，公司搞那天咱想要啦。
这些机器、这些东西根本都是武钢讲的，是真会怕别人。我求你，你不要动他们好不好？
对，他们都是跟你10几20年的老员工呢，你真的忍心一脚把他们踢开哦？
开动！
小亮，我告诉你哦，事情麻烦了，我人在工厂哦，
加油！
你们干什么
？2月21日周日晚间10点。公视人生剧展，关老爷。
阿强，今麦看哪里会当救我？
那一瞬间回望来处，该说爱或亏欠地上的女人们，制造的未亡人，一起去还活着的鬼魂，脚踏着泪痕，藏好伤痕，但 流于向现实安稳，无价之争。
香港旺角大年初一的警民冲突后，港府和北京当局相继指控本土激进分离团体虚与滋事，各种阴谋论随之出笼。美国共和党初选激烈，不实地产大亨川普言行粗鄙，连其他选将也离斯文太远。西班牙公务员太好混，有人退休前6年从没上过班，还差点获得市府颁发服务奖章。本周6日晚间7点半，公视全球现场与您一起探索全世界。
有人说，跟着蝴蝶能找到花朵，在高雄茂林，跟着紫斑蝶看见绿宝新气象。
我们先推减药，请农友就是在开花后、开花期、开花后，尽量把农药用到最少。
农民转型，紫蝶安心。2月22号星期一晚间10点，我们的岛蝶舞茂林。
生态摄影家陈维仓以78岁的行动力与生命力，数度前往非洲与南北极，谱写出《魅力非洲与纵横极地》摄影作品集，记录珍贵的环境影像及生态故事。捐款加入公视之友，获赠陈维仓摄影作品集。详情可扫描 QR Code 公视网站或拨02-2633-2000-9。
才用得吃啊？
为什么？
因为天公伯啊，给咱保庇啊。
是阿嬷煮的，又不是老天爷。
哎呦，较早若讲少一碗饭，咱就爱行人一斗米。
有，老师有说要知恩图报。
反正做人都要知影感恩，这社会唔才有温暖。
那感谢老天爷给我一个最棒的阿嬷。
公视表演厅，2月20号周六晚间11点，再续《情缘2》。
你不要觉得他是这样慵懒哦，他都有听得清楚你在讲什么。
对，就是很自在，不管怎么唱都对，都好听，都
你不要故意学他的 tempo， 不像。
每周日晚上8点，
公共电视13频道，
音乐万万岁3。
您现在收看的是公共电视，透过公视，我们看见文化，看见艺术，看见更好的未来。